Now I would like to introduce Dr. Andrew Nelson, Assistant Professor in the Department of Laboratory Medicine and Pathology.
Thank you so much, Maureen, and thank you all for being here today. It's really kind of a privilege and an honor to share with you our story about how we were able to build a clinical test for SARS-CoV-2 or COVID-19, a clinical diagnostic test in a mere 5 days. It was a major multidisciplinary effort that took input from, I think, no less than about 4 or 5 different departments and units. And really, this is actually the first time that I've had this opportunity to share the story from start to finish with a group of our colleagues, friends, and alumni. So I really appreciate the opportunity to be here today. So as I said, this will be a largely untold story of success and how we came together across these 4 departments: laboratory medicine and pathology, the University of Minnesota Genomics Center, microbiology and infectious disease department within the University of Minnesota, as well as our colleagues at mHealth Fairview and my laboratory, the Molecular Diagnostics Laboratory, MDL as I'll call it today, as well as the Infectious Disease Diagnostic Laboratory. We'll also talk briefly about why we got in the situation we did, where we had to rely on a laboratory developed test for COVID-19 that really provided about 90 to 95% of our test capacity over the last 6 months, not only for the University of Minnesota and mHealth Fairview, but actually for about 1/3 of the total state's testing capacity. And we'll basically end with a couple of vignettes, a couple of ideas about how this flexible and high performance test was able to adapt over time, how we were able to scale it to meet the needs of Minnesota as far as clinical diagnostics, and also talk a little bit about some of the scholarship and some of the academic activity that came out of it as well. So I'll break it into about 5 parts. We'll start with part 1, which was the rise to the challenge. We'll go all the way back to the early months of 2020, which I know has been a long year for all of us. And just kind of go through the crash course of the first 5 days and how we were able to build a test that quickly. You'll see that this date, 3/19, means a lot. This was a comic that I clipped out of the political commentary section of the Pioneer Press. It came in a very important time when we were going through a lot of stress and a lot of hard work to get this test up as quickly as possible. So going back to January, February, and March of this year, of course, it was a very uncertain time. We all knew that there was this new emerging infectious disease coming out of Asia, that it was really unclear how it was going to impact our world. It was really unclear how quickly it was going to spread. And while there were some predictions that we get to the state that we are in right now with the number of deaths, the number of cases worldwide, there was a lot of uncertainty. We could have gone much higher, and in some cases, we could have gone much lower. During January, February, and March, the Centers for Disease Control tried to essentially maintain control over all testing for SARS-CoV-2 in the United States of America. This is pretty much common practice for how they address emerging pandemics, new emerging infectious diseases, wanting to make sure that they have really high control, priority, and quality control over the tests so that they can get the best data in the most rapid and efficient fashion. The problem was, of course, that there was a little bit of a stumble in how they designed their assay, and it actually really wasn't so much in the design as much as it was in the manufacture of the assay. So as they deployed their assay to state departments of health across the country, there are issues with some of the positive and negative controls, some of the reagents that led to significant cross-contamination. And so we actually kind of got into kind of a negative hole with our ability to test across the country early on as the virus started spreading into North America. In light of that, the FDA went through a pretty significant regulatory shift. And so as early as February 4th, testing or state health department centralized testing was not going to be sufficient to address the emerging pandemic. They started releasing a number of new regulatory guidelines on how different types of laboratories, both commercial device manufacturers as well as high complexity CLIA validated laboratories, could implement testing under what is known as an emergency use authorization, or an EUA, as I'll call it throughout the test. Now, there was a lot of shifts, and it was very trying to try and keep up with what those changing regulations were in the early days. The University of Washington is a good example, as the first academic laboratory that developed an LDT, or a laboratory developed test. They faced a lot of roadblocks early on because, again, there was concerns over quality and accuracy in testing, which obviously is important, but they didn't want to allow laboratories that had already demonstrated competency with CLIA validation as a high complexity laboratory to bring up these types of tests. They didn't want to release that very early on to allow them to move quickly and address the need. Some of the other problems that we ran into early on in the response to the pandemic is that commercial device and test manufacturers were similarly bringing up their tests, following FDA regulations to meet high standards of quality and accuracy. But they were under the gun as well because they had essentially hundreds, if not thousands, of medical centers across the country seeking to get as many tests as possible from these manufacturers as they could. And as many of us know, oftentimes in advertising and sales, there's a lot of overpromise and underdeliver. And that was, of course, something that happened to the University of Minnesota and M Health. We were trying to figure out a way to ensure that there were adequate testing for SARS-CoV-2 during the months of February and March. During this February period, we essentially were working with two commercial manufacturers that were already, their instruments were already routinely used in the infectious disease diagnostics lab. And we were told that we would be getting a minimum of 1,000 to 2,000 tests per day. And that amount of volume never actually materialized. And that's why it became a problem and one increase. It's clear that decentralized rapid testing was going to be a need. Outside entities in the state of Minnesota basically pressed onto the university that they needed to rapidly increase testing capacity. So on March 16th, those voices were heard by leadership in the university. And on March 17th, Dean Jacob Toller challenged lab medicine and pathology to develop a test as quickly as possible. Literally, Jacob wanted it the next day. Of course, that wasn't quite possible, but we didn't do too bad. We were able to get it in 5 total days from March 17th. And so on March 17th and then on March 18th, leadership within lab medicine and pathology developed a team. We reached out to our partners that I'll tell you about on the next slide, and we started the development process for the test. So I have to give a lot of credit to my colleague, Dr. Sophia Yohe, who is the medical director of the molecular diagnostics lab at M Health Fairview and also serves as the medical director for the CLIA clinical processes that occur within the University of Minnesota Genomics Center, which is led by Dr. Kenny Beckman. So basically, the three of us, myself, Sophia, and Kenny, formed a triumvirate of leadership, where Sophia was essentially the lead doc who did a lot of the communication and high-level logistical organization with both Jacob and Tim Schacker from the medical school, as well as our leadership in lab medicine and pathology, Tony Colleen and Leo Furcht, and how they interfaced with the University of Minnesota physicians. Kenny and I were really the boots on the ground leadership that worked with components of UMGC, as well as our clinical molecular diagnostics lab, to staff personnel across this complicated org chart to ensure that we could develop the test, figure out how to handle sample intake and extraction, figure out how to get the test signed out and samples managed through the laboratory through our lab information management system. And then finally, as I said, get those tests signed out into the medical record and returned into clinicians' hands. Again, with the idea that we were all going to be able to do this, our goals and our turnaround time for testing was 24 to 36 hours, which was going to far eclipse the average turnaround times in this early period, which were on the order of 5 to 7 days. We had to do this in the context where supplies were extraordinarily limited, and I'll talk about that in the subsequent slides. But again, we had to have key personnel who were involved solely, essentially 12 hours a day, in ensuring that we had a supply chain to keep these tests going. So how do we build a test in 5 days? The first thing that we needed to do was really figure out where this space was going to be. And this lab truly ended up being, because this was a brand new test and couldn't supersede existing spaces, and also needed new levels of safety equipment that really weren't necessarily in high supply within the current clinical laboratories. We had to actually take over research space in the microbiology and research facility, or MRF building, and partner that with the high-tech genomic capacity of the University of Minnesota Genomics Center. As I said, safety was a big priority. This was a very complex virus. We didn't understand probably still don't to this day understand all of the individual impacts that the virus can have. And so we had to work closely with our colleagues in microbiology and infectious diseases to develop the best safety protocols and again, find the adequate space with the appropriate amount of biosafety cabinets to do this work in. Supplies and equipment was a massive challenge, which I will talk about in more detail as we go through the talk. And then we needed to be able to do this again at a CLIA validated level. And so usually when we run a CLIA validation for a laboratory developed test, we plan on a process of about 5 to 7 months. That's actually even after the time point where we essentially strategically develop the test and conceptualize it. So that's really the boots on the ground, doing experiments, writing up data, submitting paperwork, reviewing that, signing off on it. And so we needed to be able to do this in 5 days, and we had to do it in 3 experiments. I have an asterisk there because it actually was 4. We'll talk about that a little bit later too. We also had to get personnel. We needed all hands on deck from a number of different laboratories. In some ways, it was a blessing that this was the point in time when all of our institutions, many of our laboratories were going into a shutdown mode due to the pandemic and the emergency response. Many people would otherwise be furloughed. Normal clinical volumes were way down, and that allowed us to pull clinical personnel, laboratory personnel from numerous different laboratories to create this huge staff that needed to run this test. And finally, we had to do a lot to create both a lab information management system to track and result the specimens through our laboratory, as well as do a lot of logistics for how samples were going to get to us and how we were going to get results back out into the medical record. The earliest challenges that we had to face were sourcing supplies. At this point in time, there really were no options to get prepackaged, previously validated clinical tests for COVID-19. And so we had to use research use only chemicals and supplies and put those together and perform our own clinical validation in order to meet this need. We had to use a number of different methods to extract nucleic acids from the samples because early on, again, one of the biggest bottlenecks across the country to doing increased testing was actually the ability to process the nasopharyngeal samples into a nucleic acid sample that could be run on a PCR molecular test. We also had to be able to find reliable standards so that we could really well characterize the performance of our assay in a short amount of time and create positive controls so that we could have ongoing quality improvement and quality assurance over time as we ran the hundreds of thousands of tests that we ended up running. Finally, we needed clinical validation samples. And to here, I must give a great deal of thanks to the Minnesota Department of Health. They were instrumental in us getting hands on early positive and negative clinical samples that they were running when the MDH was the only clinical diagnostic test in the state of Minnesota. And that was key for us to really get our assay up and running quickly, because many of these other commercial manufacturers, many other even commercial laboratories were using synthetically created samples, basically faux clinical samples where they took negative clinical matrices and spiked in virus that they were either culturing or receiving from other labs that were culturing them. And in a shorter period of time, we really didn't have the option to do that ourselves. That would have taken weeks to develop, but MDH came through and allowed us to be able to quickly validate our assay and get up and running within this 5-day period. As I said, another major challenge was creating informatics and workflows. So anytime that you run a clinical laboratory, there's a great deal of work that goes into accessioning a sample, making sure that that stays tracked and linked to a patient identity, tracking that sample throughout an entire multi-step workflow, and then getting that data analyzed and formatted into a clinical report that then can cross, frankly, across numerous different electronic medical records across all of the systems within the state. So we needed to rely a lot on both internal development, which was done by the director of the UMGC, Dr. Kenny Beckman, and many of his other staff that really created the internal laboratory workflow. And then, as I'll talk a little bit later, also it required a great deal of work on the part of Sophia, my colleague, who is the medical director, as well as Shannon Gascon and Wendy Walters in Fairview, who worked with Fairview IT to create all of those interfaces and all of those connections that was needed to be able to receive specimens, trace them, report them, all within, again, 24 to 36 hours. Finally, one of the last and most important early challenges was ensuring personal safety. So again, this is a time where it was not clear what the appropriate biosafety level and the hazards were around this virus. I'm sure, as everybody remembers on this webinar, there was a tremendous amount of uncertainty, a tremendous amount of fear. There still remains that, to be honest. And so we needed to work with Dr. Tyler Bold and Dr. Ryan Langlois. Tyler is an infectious disease doctor. Ryan is a basic scientist and a virologist in the microbiology department, to really come up with very stringent safety procedures to keep our lab personnel safe. So we operated at a level of personal protection and personal safety and workflow that was really not heard of in normal clinical laboratory workflows, even within the infectious disease laboratory, which was, of course, used to working with infectious pathogens. So how did we gather this arsenal? The first couple of days after March 18th, really, I spent most of my life running back and forth across campus and across town, gathering reagents, gathering samples. Up here in this picture I snapped as I loaded boxes into the back of my truck. These samples came from the Minnesota Department of Health. This was the first set of clinical positive and negatives that we used for our clinical validation. And of course, as I wrote on it, this was pure gold and allowed us to do what we could as quickly as possible. As you can imagine, it was a little bit of a stressful time. So some of the lab staff had to leave a nice joke for me and to drop off some more reagents. They said, I snapped a picture of this where they said, please leave for Andy to keep him cool. Because as you can imagine, we were in pretty much a race where we were spending 14 to 18 hours a day over those first 5 days, doing experiments, doing planning, sourcing reagents, sourcing personal protective equipment. We got to the point where we were actually looking for donations from across the entire campus and the community for PPE, as well as for some of the extraction reagents that we were needed to building the kit. And finally, I have to give a lot of credit to Kenny Beckin's family. A couple of his children actually drove in a race all the way across to the Promega distribution center, which is outside of Madison, Wisconsin. In order to get some of the chemicals we needed to build the test, they literally drove overnight to get there and get those reagents and get them back to us as fast as possible, because that was even going to be faster than overnight shipping. So it was quite a race in those first couple of days. So how did we build a test in three experiments? So as I said, we had to very carefully plan this approach and make sure that we could do this quickly, effectively, and at a high level of quality. So the first experiment that we planned was to characterize and verify the assay that we designed. And again, the assay that we designed was based on the similar designs that the CDC based their design on, but we used entirely different sets of reagents so that we didn't run into any supply chain limitations. We had to find multiple positive and negative controls and be able to source those within 24 to 48 hours from different sources such as BEI and other repositories of pathogens. So we ran a whole slew of positive controls, including the first isolates that came from Washington State when COVID-19 actually hit into the United States. We included negative controls that included SARS-CoV-1, or what was originally known as the SARS breakout, as well as MERS. Again, these are all related coronaviruses that are similar to SARS-CoV-2, but are genomically distinct enough that we needed to make sure that the assay was specific to SARS-CoV-2, i.e. COVID-19. We needed to do linearity and range finding with the assay to understand how sensitive it was, what its limit of detection was, so we could do more formal and more focused assays in the second experiment. And we also developed what I called a compact assay format. So we were very keen on developing an assay that had high throughput and minimized the use of reagents, because again, we were at a point where supply chains were incredibly uncertain, incredibly limited, and we could not necessarily guarantee that we would be able to get all of the chemicals we needed to keep the test running. So we wanted to be very efficient in how much assay we used for each sample, how much assay components we used for each sample. Second experiment that we performed was a lower limit of detection and a precision assay. So again, precision is the idea of how repeatable the test is. So we did lots of replicates to assure that the test would work in different techs' hands and that we would get the same result with the same underlying nucleic acid sample over and over again. We had to find the correct standard, and that's a funny term that we'll get into later because, again, one of the things that has been very, I think, challenging for the general public to understand is that all of the individual assays that came out and received emergency use authorization have actually had um, very different standards that they used in order to create their performance characteristics. And I think that that has also led to some uncertainty and confusion within the field. And then the third experiment that we performed was an analytic accuracy study on the clinical samples that came from MDH, where we had 100% concordance with their samples that they provided. So we knew at that point we were able to get out of the gate. To just kind of give you a quick highlight into how complicated a lab information management system can be and what the scope of this is when you start to run in the neighborhood of 5,000 to 6,000 samples a day. Each one of the samples comes in a tube, and those tubes can be of very different sizes. And so this is a very manual process where these nasopharyngeal samples can come in a variety of sized sample tubes. We need to get those with human effort into a computer system where they can be tracked by barcoded labels. And then we need to rack those out into more standardized assay tubes so that we can start the process of viral inactivation and extraction on either manual or semi-automated instruments. So basically, we need to be able to keep all of that straight as this goes through multiple different manual handling steps, goes through some automated instrumentation steps. And then from those extraction processes, we then need to plate each small batch of 16 individually processed samples into multiple batches to get to a total of 96, which then runs through into our PCR instrument, which I'll show you on this next slide. And so we had to create a framework within FileMaker Pro, again, within 4 days to track and trace this throughout the workflow and manage the workflow as efficiently as possible. Then once the samples were extracted and plated out into batches of 96, we then did further pipetting into the assay, which is just a standard quantitative reverse transcriptase PCR assay that generates amplification curves that are specific either in the blue and the yellow to two different targets within the SARS-CoV-2 genome, or in the green, a target within human genome that serves as an internal control. And then the other half of what our internal lab information management system needed to do is able to visualize this data. And this is a real graphic that you can see of this FileMaker Pro output that allowed us as pathologists to review the results in context so we understand if there's any aspects for cross-contamination. We can look at the primary curves from the data and ensure that there's no artifacts or any other technical issues leading to false positives or potentially to false negatives. And so the timeline of how we got this test out the door in 5 days is such as you see in the upper right. Again, the scoping and the charge from Dean Toller came on the 17th of March. And the 18th, development started in earnest, and this was a lot of planning and a lot of reagent acquisition. On the 19th, experiment number 1 failed. So that first range finding experiment and positive and negative control experiment failed. And that was again because we had used some standards that we weren't quite sure of all of their performance and handling characteristics. But that created a lot of trepidation and a lot of heartache. So that was a tough night, I gotta be honest with you. But luckily, we showed up bright and early the next day on the 20th. We repeated experiment number 1, our range finding experiment, and our positive and negative control experiment. And the second version of that was a great success, and everyone was elated with that. And we were then quickly able to transition into experiments 3 and 4, which were our limit of detection determination on our clinical analytic sensitivity accuracy assessments, which were both successful. And so on the 22nd of March, we went live with clinical testing, again, in 5 days' time. Shortly thereafter, about a week, we were published a preprint paper on our method so that we could try and share that with as many other colleagues in academic laboratories, high complexity laboratories. As you can see, I had a notebook where I was taking notes on the 20th when that first experiment Number two came through. Thank God it works. I was the only thing I wrote down that day because I didn't have time to write much else. And then, as I said, on day 5, when we ran our concordance experiments, we had 100% concordance. So this was a pretty high time for our laboratory. We ultimately ended up with a limit of detection, which is very sensitive, with about 1,500 copies of the virus per milliliter of sample provided. We'll get more into the context of what that means as we go down the line. So going out of the gate, we got through this race of 5 days. We created an effective test, but it was actually somewhat slow and awkward. So we were already pretty stressed out from the validation procedure. The increased safety workflows were definitely hard on people to get used to. And there was a lot of disconnect between reality and expectations. There was basically a need for thousands of tests a day, but an inability to collect more than a couple hundred tests a day. And so it was really uncertain early on, and it created a lot of stress as far as how could we actually ramp up to meet the clear testing needs in the community. There were some informatics glitches. We had our first miss early on where there was a sample mix-up due to some informatics problems within that lab information system. Luckily, no patient harm became of that first miss, but again, it allowed us in this slow and awkward time to ensure some data fidelity and become more comfortable with our brand new workflow through some quality improvement processes when we were not testing many more than somewhere between 50 and 150 tests per day. Another part of our emergency use authorization is we needed to continue submitting samples to the MDH in parallel as we were releasing clinical results in order to do a post go live validation and quality assurance program as part of the FDA's EUA program. And that's where I say, quote unquote, the second mistake, question mark. In fact, actually what this was a patient who was very ill with COVID-19, who was in the intensive care unit on a ventilator. That patient had a nasopharyngeal swab, which we had as a low positive on our assay. And when we sent that sample to the MDH, they called it as negative. And so this created a lot of concern because this patient was hospitalized on a ventilator with COVID-19. So we actually acquired a tracheal aspirate sample from that patient. And so this sample then, is now represents a lower respiratory sample, and it was a very strong positive. We sent that sample on to MDH, and they again also got a very strong positive. And so very on, in this essentially first week of testing in March 23rd, 24th, 25th, we got my first dose of how weird and wicked a disease COVID-19 can be, where the testing, a lot of it, as far as how sensitive and how accurate the testing may have as much to do with where we sample our patients versus what are the underlying performance characteristics of the assays. As I'm sure everybody on the call knows, there was leadership breakdowns across our globe, across our nation, across our state, but it was an institutional problem as well. It was very difficult to get good communication across the disparate parts of the mHealth Fairview system so that we could have efficient modes of getting results returned in a timely fashion to the clinicians, both within the historical Fairview system as well as the Health East system. It has always been and continues to be a challenge to collect the appropriate number of samples consistent with the amount of tests available. For most of the last 6 months, there has been latent test capacity. And what I mean by that is that there have been more assays available than tests collected. And that's because it is an extraordinarily difficult logistic challenge for an institution to staff testing, drive-through sample collection facilities, et cetera, and make sure that they are collecting just enough samples to use up all of the available tests rather than overwhelming the test capacity or vice versa, collecting too few tests and having latent test capacity. There was a lot of confusion about which physicians could order and which patients they could order the tests on. And there was also difficulty because we were using 3 different platforms simultaneously, so routing tests was a big challenge. So what did that mean? Well, it meant that we were put on a 4-week resource supply allocation rule, which ultimately meant that in that first 5 weeks, we were only allowed to test up to 328 tests per day. Again, because of the inability to collect tests effectively and efficiently matched to the number of tests available, there were days where we performed as few as 32 tests. And that was, again, because the system, from a management standpoint, was limiting the number of patients or the types of patients who could qualify for a test. They were basically creating stores of tests thinking that a tsunami was coming. And there was a thinking process that was going on that if you couldn't offer 2,000 tests, there was no reason to do 600 or 800. We were limited to essentially doing 200 tests per day. We also had challenges with getting enough swabs and enough medium to collect the test, let alone having enough reagents to perform the test. But we were able to overcome that slow period. Despite the fact that nobody cares, we kept working harder, and we were able to ramp up our capacity. And this was done in part with a partnership with the state of Minnesota and the Mayo Clinic. So as I said, that low volume was a mixed blessing. It facilitated optimization of our processes to become more efficient, more accurate. We learned a lot from the test. As I mentioned, with our post-surveillance period for our emergency use authorization, we learned a lot from that one patient as far as where the disease may have the highest level of viral replication in different phases, whether it's early symptomatic phase or whether it's late symptomatic phase with severe systemic inflammatory responses. So we learned a lot about the anatomic variability. We learned a lot about the performance of the test because otherwise, again, we continued to have 100% accuracy with MDH in that post-surveillance period beside this one quote unquote miss that was actually due to the biologic variability of the disease. We also learned a lot for the potential for cross-contamination. And to put that in context, this is an assay that is sensitive enough that literally can detect one copy of the virus in a single microliter, i.e. 1,000 copies of the virus in a milliliter of primary sample. Yet we had some of our positive samples that had upwards of 30 million copies of the virus per microliter. So that's 30 billion copies per milliliter. So that is a huge dynamic range and it creates a huge potential for cross-contamination. So we had to come up with a lot of QA perspectives in order to be able to address that. So as I said, we ended up supporting the state of Minnesota. Governor Walz's administration reached out to the University of Minnesota and the Mayo Clinic to devise this testing command center. The Minnesota Department of Health recruited 10 different hospital academic entities, which included both the University of Minnesota, the mHealth Fairview Health System, Mayo, and numerous other health systems to create a statewide, more integrated, I wouldn't say fully integrated, but more integrated system with the goal of providing up to 20,000 overflow tests per day. And the bulk of that was going to be provided by the University of Minnesota and the Mayo Clinic. In order to meet that demand, we had to go through essentially continuous cycles of validation throughout all of April, May, and June to increase our capacity. In order to do that, we used a number of different types of more sophisticated automated extraction and PCR setup instruments. And we had to take each one of those through an individual or essentially a bridging study to provide additional clinical validation of the performance. We also had to dramatically increase the size of our staff. And so hiring in the face of a pandemic that has otherwise shut down pretty much all economic activity is quite challenging. That state-funded investment led to the purchase of, again, more sophisticated automated instrumentation that allowed us to increase our volume. But again, as I said right here, some of this was finicky. So again, we were working many long hours in the background doing essentially continuous research and validation work in order to increase the capacity and meet the state's need. So in April, the University of Minnesota went live with testing for this state testing command center program where we were doing at that first phase between 2,000 and 4,000 tests per day to meet the state's needs. We needed to increase our ability to have informatics interface with the Mayo system because Mayo's statewide collection system was providing the backbone on how to get samples literally from every county in the state of Minnesota, either to the Mayo medical labs or to the University of Minnesota for extraction. And finally, when we hit our stride in June 26 with those most sophisticated instruments provided by the state grants, we were able to really meet our final goal of providing somewhere between 5,000 to 7,000 tests per day for the state testing capacity. So over this period, what were our clinical accomplishments? So as I said, we were a true pop-up lab. This is the microbiology research facility, which is on the backside of the technology districts, on the other side of the stadium on the University of Minnesota campus. It was completely shut down except for us operating that first floor. These are my two kids that provided a lot of emotional support for our lab. I'm sure many of you saw all of the great work that people were doing to support our colleagues at Bethesda Hospital. And so they came and did the same thing for us. So how did our volume growth accelerate due to the state testing program? As I was telling you, that early phase in March, we were doing very few samples per day. So our volume in 2 weeks in March was 1,000 samples. Our volume in the entire month of April was only 3,000 samples. But once the state testing command program took place on April 24th, went live, in the month of May, we did 36,000 samples, essentially a log-fold increase in our volume. And then in the month of June, we went on to do 115,000 samples. This is a graph that shows that volume over time, and it really takes hold at how important this laboratory developed test was in comparison with the commercial assays that were available. So in blue, you see the volumes of tests on a per day basis that were performed on the laboratory developed test at the University of Minnesota. In contrast, these are the number of assays that were performed in the mHealth Fairview Infectious Disease Laboratory on the two different commercial platforms that we had available to us. Not a single day did they ever get above 500 samples tested because of severe supply limitations on those commercial assays. On the other hand, we were able to have sustained volumes between 3,000 and 5,000 with peaks of up to 7,000 during the life cycle of the test as I'm presenting it to you, which basically ends at this red line on September 1. I'll talk a little bit more about what happened after that as we move on. So this gives you an idea of the results on a bi-week basis. You can see in gray down below what were the total number per week of positive tests that we reported. And as you can see, this essentially mirrors the amount of positive tests that were reported across the state of Minnesota because we were providing about 1/3 of the state's total testing volume. And you can see just how significantly our volumes increased on a per week basis over this period of time. And again, when you take a close look at that relationship of the number of positive samples per week to the total number of volume per week, what you could see was that early on when there was low test volume, we were not anywhere near to testing the appropriate number of people in the community. We had a very high test positivity rate between 7 and 16%. But once we got up to sustained volumes on a per week basis of 20 to 45,000 tests per week, we got to much lower positivity rates, generally between 2 and 9%, and predominantly below 5%, which is where the state epidemiologists want us to be in order to ensure that we're testing at least an appropriate number of symptomatic people to help them keep good track and tabs on how the disease is spreading throughout the state and help them manage it. So it was a good run for us. So this pop-up laboratory ran from March 22nd to August 31st. As the University of Minnesota started to reopen its research enterprise, we needed to vacate that space because there's grant-funded research that normally was going on there. And so we needed to move that test to a new home. As we moved that test to a new home, we moved it to a different component of lab medicine and pathology called the Advanced Research Diagnostics Lab. I'll have a few words on that in a couple of slides. But overall, during this period of time from March 22nd to August 31st, the laboratory at the Microbiology Research Facility, UMGC, signed out a total of 267,000 samples, which was an average of approximately 1,650 tests per day with an overall positivity rate over the entire life cycle of the test as it was there of 3.5%. And this was our all-time signed out high score, which is a little bit less than what I showed you, is the number of samples processed of 6,547 tests released in a single day. So as we transitioned to Ortal, we needed to move over all of that automation equipment. We developed validated a new but related PCR assay. This was a commercial product, but it was a commercial product that was intended for only high complexity laboratories, and therefore there was not a significant supply chain limitation on it. It used the same targets as the initial assay that we developed, but had higher throughput. So what that helped us do is it allowed us to apply the many lessons that we learned in those first 4 months over at the MRF UMGC laboratory, make sure that we didn't fall into the same pitfalls, and again, develop flexibility in our supply chain and our approaches so that we continue to grow our volume. And as you saw with the University of Minnesota graph of the number of tests performed, we've continued to accelerate that number of tests performed on a daily basis to an average between 6,000 and 8,000 tests per day. So in that context, too, again, we've been able to actually not only provide this level of clinical care for our patients and for the state of Minnesota, but we've also been able to actually do some academic investigation as well. As I said, quality control was a very important part of this process. I will mention that in some ways, I think some of the molecular tests are so sensitive that false positives are as much of a risk as false negative testing. And really where that can come from is environmental contamination. Even the best of laboratory practices can still lead to trace amounts of aerosolization when you're using a PCR amplification technique. As little as one molecule can create a false positive signal if it contaminates a large sample. So we actually had a fantastic resident in our program, Ryan Martinez, who looked at our different methods of preparing our samples. We had manual extraction methods. We had 2 different automated extraction methods. And he developed different types of models that allowed us to understand how could you get contamination from a well of interest into potentially surrounding wells. So again, going back to my analogy, if this well has a concentration of 10 million virus particles per microliter, but in this well, I can detect as little as 1 virus particle per microliter. We need to have very robust ways and automated methods that we can develop in order to ensure that we repeat those samples and provide the highest accuracy and quality in our results. And so he developed a couple of different models. Again, I'm not going to go much into the details and the data itself in the interest of time, but he developed computational models that looked at different characteristics of the total amount of relative viral load within our different extraction batches, within the differences in the relative amount of virus between parallel wells or neighboring wells, in order to come up with great quality assurance models that helped us to prevent false positives. We also performed assay equivalency. As I mentioned, we were testing on 3 different platforms throughout the entire life cycle of this assay, and that practice again continues. Even though the laboratory developed test has this huge high throughput scalability, it is a little bit slower, whereas the commercial diagnostic tests, which are the diasorin and the cepheid, although we only have about 500 of those assays a day, we can turn those tests around within 2 hours. So we've had to develop strategies where we prioritize tests for hospital admissions and those types of situations where we need the most rapid turnaround measured in a matter of minutes to hours rather than a matter of hours to days. And so we needed to know what the equivalents of those assays were. So we were able to do some cross-comparison studies where we compared 30 positive and 15 negative assays from the diasorin to the laboratory developed tests and from the cepheid to the laboratory developed tests. What this data shows you is it has our cycle threshold values. For those of you who may not be totally up to date on your PCR and your molecular pathology, the higher the value, the lower the amount of virus in the sample. What we see here in the circles are the tests that were not necessarily concordant in the pairwise comparisons. And what this tells you is when you have cycle threshold values of 35 or greater in your discordance, where we had less than perfect agreement was in the range where, again, we're talking about a few copies of the virus per microliter or even a few copies of the virus per milliliter. Overall, the agreement between the platforms was very good. The LDT had a 98% overall agreement with the diasorin and 93% overall agreement with the cepheid. So we knew that we could largely use these assays interchangeably. But again, the question is, are some of these PCR tests too sensitive? We knew that the discrepancies were at the level basically at the limit of detection on the assays. The Cepheid was the most sensitive. Therefore, that was why there was the lowest overall agreement there. The problem is, again, that when we talk about what has the FDA has approved for COVID testing, the entire range is gigantic. So our assay was at 1,500 copies per mil. The Cepheid assay was at 250 copies per mil. The diasorin was at 500 copies per mil, and that largely explains the small amounts of discrepancies that we saw between the assays. But the range of approved assays that the FDA has granted EUA to has literally gone between 100 and 180,000 copies per mil. So again, that can probably provide some context to why you've seen so much confusion and uncertainty in the public press about why results may not be consistent between different types of COVID tests. We've also undertaken a project to validate saliva collection. You've probably heard in the media that the state has separately signed a contract with an outside company that was a spinoff from Rutgers University to provide saliva testing. We are also in the process of rolling that out within the University of Minnesota. But we've also put this together into a developing manuscript with my colleagues Dan Knight and Abby Johnson. And really what this shows you up here, we had two different types of cohorts. Cohort 1 was essentially a screening cohort. So these were patients who are presenting early on in the course of disease. And cohort 2 was actually patients who were hospitalized in later parts of the disease while they were at Bethesda. And what we found out again is that there is significant anatomic variability to whether we can detect virus in a nasopharyngeal sample versus an anterior nasal swab sample versus a saliva sample. Overall, the concordance is quite good, particularly in the early screening phase. There were only 2 misses. And in fact, these 2 patients actually were not in a screening phase. They had been previously diagnosed with COVID-19 about 2 to 4 weeks prior, and they maintained having a high symptomatology index because, again, they fell into this relatively poorly defined category of long COVID patients, and they were being rescreened to see if they had a large amount of virus still present, and they had virus that was detectable within their nasopharyngeal specimens, but not in their saliva or not in their nasal samples. Otherwise, particularly in the screening population, if we exclude those 2 patients, we had 100% concordance. So we feel very confident that saliva is a very good sample for screening patients to make a diagnosis of COVID-19. Hopefully many of you had a chance to catch Chris Tignanelli's previous presentation. We had an opportunity to support Chris and Mike's trial to look at the utility of losartan as a drug that could potentially mitigate and minimize the symptomatology of COVID-19. That's been a very complex and challenging trial, and it required CLIA-grade PCR testing of patients multiple times. Each patient was going to get up to 5 tests over the course of the treatment from day 1 to day 15 after their diagnosis. And as you can no doubt appreciate, with the severe supply limitations of any of the commercial testing, our laboratory developed test was the only option that had the scalability and the robust performance to meet this need. So we've had the privilege to work with them and support the functioning of that clinical trial over its entire lifetime. So thanks to my colleague, Dr. Yohi, who created these medals for everybody that was involved in the testing, with live and 5 being kind of our motto. But what are some of the lessons learned that I'll leave you with, and hopefully we have at least 5 or 6 minutes for some discussion. Analytic sensitivity, biologic heterogeneity, and clinical utility are different concepts. So hopefully one of the things, and happy to answer questions on this, that we can talk about, many of the PCR tests out there for COVID-19 have very high analytic sensitivity. The differences in results, the questions of false positives or what is the clinical sensitivity, I think is largely driven by biologic heterogeneity. And on the flip side of that, I think that some of the tests are actually so sensitive, they're detecting excruciatingly small amounts of the virus that people may carry for a short period of time. And there's emerging data that suggests that those patients don't actually have significant amounts of live culturable virus in their system. They're very unlikely to be infectious. So there's still just a great deal of uncertainty about the clinical behavior of this of this disease. And I think overall, all of the assays that have tried to address the pandemic are very good, but there's just a lot of uncertainty in how we clinically apply them. Again, false positives in that same vein and false negatives create concern. It's a different type of concern, but I think it's equally important. Really, what has been the biggest challenge for creating sustainable and scalable testing is the system logistics and getting the personnel to just continue to do this scope of work. Again, putting it in context, we signed out nearly 270,000 tests in 4 months for a test that did not exist the day before we started. Finally, I think that there's a gap in communication and knowledge in the face of uncertainty. I think that there's been a lot of pressure and not a lot of time for laboratorians to communicate test performance issues, sample site anatomic variability, these types of things that I'm telling you about out to our frontline colleagues. And it's been an extraordinarily challenging time for those who are in charge of the hospital systems trying to figure out how to collect, process, and route all of the tests, all of the samples that need to get to the testing capacity that exists. And hopefully that can continue to get ironed out over time so that we can continue to increase the overall testing capacity for the state and continue to try and get a better handle on this terrible disease. I'd like to leave some interest, in the interest of time, I'd like to leave some time for discussion and questions. I won't go through all of these names, there's too many. But again, this was just a huge team effort, a multidisciplinary effort, and every single person on this list has played an important role in allowing the institution to meet this need of the state and of the hospital system. And there are many, many others in Fairview and UMGC, and other divisions that I didn't even have room to put on the slide. So with that, I'd be happy to take any questions, and thank you very much for your time and your attention.
Yeah, we'll give everybody a minute if you have any questions, if you'd like to type that using the Q&A function. I believe that we answered the question about your opinion on the saliva tests that have come out.
Yeah, and I could be happy while we're waiting for some others to provide a little bit more information for that. So again, Vault Health is the name of the company that is bringing up these labs with a contract with the state of Minnesota. Their data was generated out of Rutgers University. Again, their data looks pretty good. I'm not going to overly criticize any of my colleagues because we do the same thing. I think most on the line would appreciate that oftentimes clinical validation studies are relatively small in scope, and it's not until we have thousands upon thousands of tests performed in a live clinical setting that we really truly know the performance. But again, the data that we've generated in-house with our own version of a saliva-based test, my review of the Rutgers data, again, particularly when we're talking about providing an easily accessible sample type for people who have just become symptomatic, and need to know, is this COVID or is this not COVID, i.e. flu or another respiratory illness. I think it's a very good sample type. I think that it will significantly improve our ability as a state to address the emerging increase that we're seeing in the numbers reported by MDH right now.
Great. We have another question from Dr. Olala. What is the best way to answer the public question about what to believe on testing?
Wow, that's a tough one. And that is, I'll probably show a little bit of my own biases, but I think it's nuanced. And I think that we have to, and particularly since I assume I'm speaking to a relatively broad audience of physicians with different specialties, but I think the one thing that links all of us is that we know that there is very little that can replace a physician's intuition. There's a lot of art in medicine, and I think one of the things that I try and tell my colleagues is that there is no such thing as a perfect laboratory test. All laboratory tests have to be interpreted within the context of the individual patient's history of present illness, their symptoms, how their disease is progressing, their individual background situation and exposures. Again, I think I would say that that the tests are good, but that there's tremendous biologic variability in the disease, and that's what is fueling a lot of the uncertainty, and that physicians just need to be cognizant and transparent and open about how we all need to adapt to the nuance, the the individual details of each patient's story to understand what a particular test result, whether that's positive or negative, means. And I always think that retesting patients where the clinical story does not fit with the laboratory test is the most prudent thing. And it's been such a challenge right now because of the test limitations to do what I think physicians would normally do. Because, you know, if you're on the hospital ward and you got a serum sodium that didn't make sense, the first thing you would do is call the lab and ask them to repeat the analysis. And the second thing you do is call your nurse and have them redraw the sample and send a second one. And you would work through it using your brain. And we just haven't had that ability. And I think the lay press has done a terrible job of trying to claim that it is all about test performance when it's really about disease and understanding disease and not knowing all of the aspects of the disease. So I see a few more. I'll stop my long-winded answer on that.
We had another one, Dr. Nelson. Will there be a time when these tests are available to the public, much like a pregnancy test?
I've seen some papers out that are trying to use different types of techniques that really could be implemented in, say, you know, a Walgreens or a CVS, whether or not it would be, you know, a colorimetric test where you take it home just like a pregnancy test, whether or not it would be something that is easily administered by, you know, a pharmacy tech or an MA or whatever. I do think that we're going to get there. I think that we'll get there. I don't necessarily want to say quickly, but I do think that those are tests that we will see during the course of how we respond to the virus, and probably before we can say that we've put COVID behind us.
Great, we're just about at time, but if you can answer Dr. Frenning's question very quickly, what's your opinion of the utility of antigen tests?
I think the antigen tests really will actually be kind of that option. And so again, let's just very quickly set the stage. There's three ways to test right now that are out there. There is a molecular-based test that detects the nucleic acid of the genome of the virus. There are the antigen-based tests, which detect protein antigens of the virus in different biologic samples. And then there is the blood test for antibodies that are subsequently formed after an infection where a productive immune response. But for the antigen test, I really do think that that's going to be the appropriately sensitive, quick, and deployable screening method that we can use as point of care in all of our pediatric clinics, all of our primary care clinics, to again quickly rule in someone. So if someone comes in ill, has an antigen test, it is positive for COVID-19, the specificity of those tests are pretty good. Then the patient knows to quarantine. We know how to manage them. We know how to make the diagnosis. If that test is negative, in the office in 10 or 15 minutes, we can do an P sample and send it for a potentially more sensitive molecular PCR test to fully try and rule out the disease. But they'll have a very important role, in my opinion.
Excellent. Well, thank you so much, Dr. Nelson, for sharing the incredible work that you and your team and your colleagues have done. As people have mentioned in the Q&A, there are so many reasons to be proud of how the medical school has responded to the pandemic. And your work getting us quickly into testing is certainly among those reasons. So thank you for sharing today.
Thank you.